This is an A-level biology presentation looking at mutations. In this presentation, we're going to look at the different types of mutations. We're going to look at how mutations can arise and the causes of mutations, and then we're going to have a look at what mutations can do in terms of actual impact on polypeptides. To get started, all you have to do is describe the transcription and translation processes. Pause the presentation now, go through in as much detail as you can, transcription and translation, and when you're ready you can restart the presentation. At an absolute minimum, transcription and translation fit into the process of turning DNA and producing an RNA strand, and an RNA strand and used to produce a protein. Make sure you don't say DNA is turned into RNA. The DNA still exists at the end of the process and the RNA still exists at the end of the process. It's simply a case of one is used to produce the next one. In more detail, we can go into transcription in terms of the use of helicase to unravel two strands. We can talk, of course, about the template strand, and we can talk about complementary pairing between each of the different types of bases. Remember, if we are producing a messenger RNA strand, we will produce uracil, not thymine. We can also talk about high complementary hydrogen bonding, and we can even go on and talk about splicing. In terms of translation, we need to go through the entire process from the ribosome large and small subunit attaching to the messenger RNA strand. We need to talk about the P site and the A site. You need to talk about the movement along the messenger RNA, insertion of new anticodons from the transfer RNA, matching with complementary codons. You need to mention triplet code, you need to mention peptide bonds between amino acids and polypeptide, and you can talk about start and stop codons as well. There should be a separate presentation looking at transcription and translation. If those processes don't make sense or you miss the words I've just used, make sure you go back over those. In front of you, there are 6 pictures representing 6 different types of mutations. All you have to do is try and work out what the pictures represent. You can pause the presentation, try your best on the 6 answers, and you can also, of course, try the extension question and try and work out which of the mutations will have the biggest impact and why. So pause the presentation now. The 6 different types of mutation are: we have what was originally referred to as an insertion mutation, but you should be talking about it in terms of an addition mutation, and that's where we're inserting a sequence of nucleotides. We also have what is called a delete mutation, which is where we're removing nucleotides. We can have what is called an inversion mutation, which is where we simply swap one section so it reads in reverse. We have a substitution mutation, which is where one section is replaced with another the same length. We have a duplication mutation, which is where we have repeats of particular sections. And we have, of course, what's called a translocation mutation, which is where we have a section of DNA from somewhere else inserted. Please note, mutations all occur on DNA. We can talk about DNA bases and mutations affecting DNA bases, or mutations affecting DNA nucleotides, which then will result in changes to messenger RNA strands, which eventually could lead to changes in proteins. In terms of which one has the biggest impact, well, the biggest impact is going to be any of the mutations that results in a change of the number of nucleotides. Because if we start reading nucleotides as triplets and we change one triplet so that instead of a 3 it reads as a 2, all of a sudden we have a huge impact on every subsequent triplet. If we have a substitution, we can take 3 out, we can put 3 in, we can take 2 out, put 2 in, take 1 out, take 1 in. We will only affect one particular triplet. Whereas if we go with something like a deletion or an addition, we can actually affect large numbers of triplets by simply adding one extra base. All the other bases are out of sequence. So what you've got is the kind of question you might be asked on the exam in terms of converting DNA into different strands of RNA and then using that to identify amino acids. At this point, pause the presentation, work your way through the questions, try your best with it, and whenever you're ready, you can restart the presentation. What you may find useful is to go through the presentation once and then come back to the presentation later on in a little bit of time. Pause the presentation now. So question 1 says, list the matching amino acid sequence. Well, the sequence we're given along the top is a DNA sequence, and the sequence inside the table is RNA. So what we have to do is we have to take the DNA sequence, we know it's DNA because it's got thymine in it, and we convert it into an RNA strand. What we're then going to do is take each of those triplets on the RNA strand, the messenger RNA strand, and identify which of the amino acids we've got. And if you do that, you'll notice that this is the matching sequence. Notice we haven't written out the entire name of the amino acid, we've just written the letter in red next to the relevant amino acid. If we have a substitution to number 4, what we're going to do is we're going to get number 4, which in the messenger RNA is C, and we can replace it with any one of the other letters, and if we do that we will actually change which amino acid we have as the second amino acid. If we go in insertion, and this time we're going to have an insertion in number 18, depending on how many different bases we insert, we'll actually not change amino acid number 6, but we could have an impact on amino acid number 7 depending on how many we insert into there. In terms of a deletion mutation to nucleotide number 10, well, if we delete nucleotide number 10, what happens is that, as we said before, all the other triplets are no longer in sequence. All the other triplets break up. And as a consequence, the next triplet becomes AGA on the messenger RNA. Then we have AGU, then CGA, and we actually don't have an amino acid at the end. That last amino acid, number 7, no longer exists because we only have 2 parts of the triplet. And as a consequence, you can see it changes our amino acid sequence. If we have an inversion of 12 and 13, an inversion of 12 and 13, we still have the same number of amino acids, but we simply swap around what amino acid number 4 and 5 are in the sequence. The extension questions were, what factors would change the impact of a mutation? It comes down to where the actual mutation occurs, because a mutation at the start can have a much bigger impact than a mutation at the end of the sequence if we have what's called a frame shift. And the frame shift is what happened when we had that deletion mutation, and because we deleted, the triplets were no longer in sequence, and we from that point forward had different sets of triplets. Do all mutations have a negative impact? No, not all mutations do. Many mutations actually have no impact whatsoever. For example, anyone who has a birthmark, that is actually the result of a mutation. So how do we get mutations? Where do mutations come from? Well, mutations actually occur on a regular basis, and they are normally, as I said, they occur in places that don't actually have any negative impact at all. They're normally neutral. However, we do have substances that can increase the rate of mutation inside an organism. Again, we've got different pictures to represent each of the different mutagens. Pause the presentation, try and work out what the 5 pictures represent, and when you're ready you can restart the presentation. Don't be afraid to have a go at the extension question as well. Pause the presentation now. Well, the different pictures represent, first off, you've got your alpha, beta and gamma radiation, your ionizing radiation. We have, again, smoking. We have carcinogenic chemicals. Those are chemicals that can cause cancer. We have ultraviolet light, and we have X-rays. And all of those substances will increase the rate of mutation inside an organism. In terms of all sunbeds should be banned, I'm going to leave that one up to you. There are pros and cons for it, but certainly the more you use a sunbed, the more regular you use a sunbed, the more damage you're going to do to your skin, and certainly the more likely you are to develop different mutations and possibly even lead to cancer. I'll leave the decision whether to use it or not up to you. So last thing, these are the different things that we've just gone through. So at this point, you should be able to describe the different types of mutations. You should be able to explain how they can have impacts on amino acid and the concept of frame shift. You should be able to explain why some mutations do not result in a change. So particularly if the change is the third base in a triplet. Changing the third base in a triplet quite often will not change the amino acid, and it comes down to the concept of degenerate code. If you're not sure about degenerate code, please do go back and look over that. It's an important concept and basically relates to the fact that each amino acid can be coded for by more than one triplet. So we can have more than one set of 3 bases giving us the same amino acid. And lastly, of course, about the causes of gene mutations and the possible impact of being exposed to different substances. So just to finish off, all you have to do is make a list of the images that you've seen in this presentation, and then you can go and say, what does each image represent? Remember, there were 6 types of mutation, there were 5 types of mutagen, and then you can also include any other images that you've seen as we went through. Once you've finished, you can go back through the presentation if you're missing any and make sure you memorize all of those 11 pictures at a minimum.